Fine. Chalo, we'll start with the session where we had winded up in the last session. The lysosome structural aspect we have seen. The functional aspect we just started. Okay. So here we'll continue with the functional aspects of lysosomes. Okay. Julie, yeah, if you find the name mysterious, let Vanessa take care of the things. Yeah, we'll continue our session. Thank you. So here where we started in the last session. Okay. Now, when you see the lysosome's function, first I introduced a word polymorphism. This was the question I asked in the yesterday test also. So poly stands for many, and morphism something defines the structural aspect of something. Okay, so if you have some structure that occurs in different forms, then we call it as polymorphism. So here when you take lysosomes, the lysosomes exist in various forms. Primary lysosomes, secondary lysosomes, tertiary lysosomes, and quaternary lysosomes. These are the various forms we will find in lysosome structure. Yes, there are 3 forms and the 4th form is also there. We'll discuss now. In the last class, I introduced the whole terminology, but I discussed primary, secondary, and tertiary. Now we'll discuss the quaternary also. Okay, Ronak? Okay. Now, it gave wrong. How? In the test, it gave wrong. What was given wrong? The options or the answer?
Ma'am, the 4th degree. I thought it was not there. So
4th degree is not there.
Ma'am, in the options it was there, but I thought it was not there. So it was my mistake. Sorry.
Yeah. Anyway, you would have seen the 3 degrees, no? So if you have seen these 3 degrees, it definitely goes for polymorphism. Okay? Fine with that? Okay. All the above option is there, Kavya?
Yeah,
for this question?
the above answer was there. Yeah, there was.
Okay. Okay. Yeah. Above will be the wrong one. Okay. We'll continue with the ongoing topic. So here, what I'm saying is we are having a primary lysosomes. Okay. Primary lysosomes are the lysosomes which will have the enzymes, and the enzymes will be inactive. The hydrolytic enzymes will be inactive in primary lysosomes. So that's the specification of primary lysosome. But when the lysosomes gets fused, okay, when they get fused, with phagosome. Then they'll become secondary lysosomes. So what is phagosome? I just explained in the last session. Phagosomes are the one which are formed when any foreign particle enters, like the bacteria and all. So phagocytosis is basically related to bacteria. Can you see the marking I'm giving? So the red color one, they indicated with bacteria. So that is coming through phagocytosis process. So by phagocytosis, what are foreign particles like bacteria enters, the cell membrane will evaginate and it will pinch off to form phagosome. This phagosome then fuses with primary lysosome, that we call as secondary lysosome. Clear? Then when secondary lysosomes we take, secondary lysosomes possess active enzymes. The enzymes are active. So here in both the primary and secondary lysosomes will have enzymes, the hydrolytic enzymes, but they are active only in secondary lysosomes. Active enzymes will find only after fusion of primary lysosome with phagosome. Okay. Now, not only the phagosome fuses with primary lysosome, but also a kind of word called endosome. So in the last session, I gave endocytosis word, then pinocytosis words, if you remember. So here endo stands for taking in solid food particle and pino stands for taking in fluid food particles. So when such food particles tries to enter into the cell, the plasma membrane again evaginates like this. The plasma membrane evaginates and takes in the food particles, either solid or liquid. Endocytosis and pinocytosis. Okay. Now once they get into the cell, they will be called endosome. Ronak, endocytosis is literally taking in only solid particles like this by the plasma membrane evagination, or endocytosis we can say through receptors that we discussed in plasma membrane. Yes, through receptors also the particles will be taken in. So suppose if you take, this is a kind of receptor which is there on the plasma membrane, only the fitted type of particles like this can enter into the cell. This is also called endocytosis, but that is receptor mediated. Okay. Ronak, your doubt clear? Receptor mediated one? Endocytosis taking
Yes,
in solid particle usually through plasma membrane evagination forms endosome, and it may happen along with the fitting type of countable receptor mediated endocytosis also happens. This is another pathway. Okay. How does the phagosomes that comes in human and plant cell which doesn't have a lysosome take it? See, for, yeah, good doubt, Tavya. See, humans, one concept, plants, another concept, animals, the concept which we are reading now. Okay. Now when you take plants, we'll be having some special type of structures which we'll read under microbodies concept. Okay. So here we didn't started the topic actually. So microbodies will take up the lysosome function in plants. When you take humans, you will be having various types of cells which will take the function of lysosomes. For example, the best example everybody can understand is WBC. You might be knowing WBC. These are the blood cells, like the RBC. Yes, RBC, WBC, you know these words? Okay, yeah. So when you take WBC specifically, WBC stands for white blood corpuscles, or we can say cells, white blood cells or white blood corpuscles. What they will do is, they'll do the same function as lysosomes will do. This is one example in humans, specifically humans I'm talking. Along with WBC, we are having some cells called macrophages. They also will do the endocytosis process, or you can say the phagocytosis. The phagocytosis, taking in the bacteria and virus particles. This also will be done by macrophages. And some other cells are also there. We call them as neutrophils. So like this, we are having various types of cells in humans which will take up the process of phagocytosis or endocytosis. Like we find lysosomes in animals. So specifically we are talking about animals, so we are discussing lysosomes. Okay, when we discuss the humans, we'll discuss all these cells. White blood corpuscles, neutrophils, macrophages, and so many other cells are also involved. And when we talk about plants, microbodies, some microbodies will take up the function. So this we'll discuss today. The microbodies, different microbodies are there in plants. Those microbodies we'll discuss in today's session. Clear? Yeah. Okay. Fine. So here we were talking about the primary lysosome. Then the fusion of primary lysosome with phagosome or endosome. So get familiar with the word phagosome and endosome. Phagosome stands for when anything enters into the cell by phagocytosis. And endosome stands for the membrane pinched off to form a vacuole-like structure when the process goes by endocytosis or pinocytosis. Okay. Now, once the secondary lysosomes, active enzymes get fused with either bacterial cell or the solid or liquid food taken in, they will be digested. There is a first function, intracellular and extracellular digestion. So here the lysosome primary function is digestion. Digestion of intracellular matter and extracellular matter also. So here we are talking about the extracellular matter, isn't it? Because the bacteria coming from outside the cell. Even the solid particles are coming from outside the cell. So here the extracellular material digestion happens inside the secondary lysosome. Once the digestion completes, whatever be the unwanted or undigested material is there, everything can't be digested. Some materials can't be digested, isn't it? So such undigested material will remain in residual bodies. So residual means what? Waste. The undigested waste material will be left after digestion secondary lysosome is residual body. Okay, so residual body we call as tertiary lysosome form. Primary lysosome, secondary lysosome, then tertiary lysosome, also known as residual body. So we are having undigested material in residual body which will be sent outside of the cell, the waste material. So this process we call as exocytosis. Exo means what? The waste will be sent outside the cell. So whatever material taken inside the cell were digested in secondary lysosome, and after digestion the residual bodies which were filled with undigested or waste material will be sent out by a process called exocytosis. So, if you understand the terminology, it will be easy to understand the concept. Okay, exo. So moving it outside. So to outside, the waste material will be pushed off. This is what happens with primary lysosome, secondary lysosome, then residual body or tertiary lysosome. Now coming to the quaternary lysosome. So quaternary lysosome, generally this word we find in higher classes only. In your standard, usually they give the word autophasic vacuole or autophagosome. This is another word. So these are the different terminology. Autophase vacuole, other name quaternary lysosome or autophagosome we can say. Okay, now come to this point. Auto means what? Auto means self. So what are the material which is there inside the cell which became old? For example, you take some cells having structures like chloroplast. Just example I'm giving, chloroplast example I took because everybody know about chloroplast. Yes, chloroplast is a structure which carries out photosynthesis. Be having more than 20 to 30 chloroplast in plant cells, small plant cells if you take. So all the chloroplasts will carry out the photosynthesis. After photosynthesis, within few days, these chloroplasts will die and a new chloroplast will be produced by the cell. The best example if you take in animal cell, because we are talking about lysosome in animal cell. So I'm taking another example, animal cell. In animal cell, RBC, the red blood corpuscles, which we find in blood, their lifespan is just 120 days. So they live for 120 days. After 120 days, these cells will die off. The RBC cells will die off. So like that, so many cells will be there in animals or in plants. So here we're talking about lysosomes. So we'll take the example of animal. So in animal like RBC, you'll be having so many other cells which will die after once their life is completed. And new cells will be produced later. That's a different process. Don't bother about that. Right now, the dead cells has to be removed from the cell interior, isn't it? The old or dead cells, if they get accumulated in the cell, the other functions of the cells may get disturbed. So in order to remove the old cells or the dead cells, some of the lysosomes will take up all those cellular debris. This is the biological word. So what are the words I used so far here? Examples are all related to this word, cellular debris. Debris means what? Waste again. Okay. So within the cell, what are the old dead cells? They are called cellular debris. And this cellular debris will be taken out by some lysosomes. So such lysosomes are called autophagosomes. Autophagosomes. Auto here stands for self. So within the cell, the old cell material will be taken. Yeah. So those are called autophasic vacuoles. So this we call as autophasic vacuole with dead and old cells or the cell debris. Cellular debris, you want me to repeat, Ronak? Yeah, fine, no problem. Cellular debris, here I have given an example. We are talking about the lysosomes in animal cells, isn't it? So I'm taking animal cell as an example. Suppose in animal cell we'll be having various cell components. Among them, I took an example of RBC. Okay, these cells will form the circulatory system in animals, but they will live only for particular time period. Assume that here they live for 120 days. So after 120 days, this RBC will die. and new RBC cells will be produced. That's a different process. Okay. So once such cells are died, or any such cells die or they'll become old, such dead old cells we call as cellular debris. Debris. Debris means waste. Waste within the cell. Okay. So waste within the cell is nothing but the old cells or the dead cells. Means they have completed their function. They doesn't carry any function. They were dead or they were old, and they have to be removed from the cell for proper functioning of other organelles. So for proper functioning of the cell, these things has to be removed. So they are called cellular debris, means cell waste. Okay, clear? Okay. Now such cell waste will be taken up by called autophagic vacuole, autophagosome. Okay. Now this will again fuse with primary lysosome. So whatever digestion has to take place, fusion with primary lysosome is important. Like the secondary lysosome fuse with primary lysosome for digestion. Same way, autophagosomes will fuse with primary lysosomes, and the waste will be sent outside. Okay, by process of exocytosis again. Clear with terminology, cellular debris, autophagosome? Any doubts? Okay. Yeah, Veda. Okay, thank you. So here, this all digestion of materials which are coming from outside is extracellular digestion, and intracellular digestion is something related to the word autophagy. Autophagy stands for intracellular digestion. Autophagy means the autophagosomes are taking in the cell debris, so it's intracellular digestion. Within the cell digestion, outside material digestion. So digestion is the important function of lysosome. First, foremost, an important function of lysosome is digestion. It happens by extracellular method or intracellular method. Intracellular method, we are giving a particular term called autophagy. Eating away the cell waste. Clear with the autophagy word? If autophagy word is okay, then I'll explain the autolysis word.